Okay, chapter 3, part 3, chapter 2, part 3. Maybe we'll finish up this chapter in this one. I don't know. Okay, so moving on to our lipid molecules. So our lipids are hydrophobic. They have a high ratio of hydrogen to oxygen in their structures. They also have more calories per gram than carbohydrates, so it makes them a more energy efficient molecule for breaking it down to get the energy that our body needs. And so there's 5 primary types in humans. We have fatty acids, triglycerides, phospholipid, eicosanoids, as well as steroids. So the first type are fatty acids. They're chains of 4 to 24 carbon atoms with a carboxyl group at one end, which is this right here. And a methyl group on the other end. So you have 2 types of fatty acids. You have saturated fatty acids that have no double bonded carbon in its chain. So here we have a saturated acid here, so no double bonds, and so every bond that carbon has, it's saturated, so there's some other atom that is attached to that carbon. So it's not like there's another way to put another atom onto that chain, because carbon has all of its bonding sites used up by a different atom. So your other type are your unsaturated fatty acids, which contain double bonds in the structure, and so it basically gives you potential to add hydrogen to that structure. So some of your fatty acids can't be made by the body, and so therefore they're called essential, and therefore they must be obtained from our diet, so from food. So one way of storing fatty acids in the body is by attaching it to a glycerol. So our other type of lipid, so here's in the blue box is our glycerol molecule, and you can attach up to 3 fatty acid chains onto that glycerol molecule. So they don't have to be the same. So in this case, having 3 different fats on that molecule, just kind of pointing this one out here. This one on the far end being an unsaturated one. So here you have potential for adding hydrogen atoms because you have double bonded carbon molecules. Anyway, so triglycerides, so we have our tri for our 3 lipids, and then our glyca part for the glycerol molecule. So each of these bonds is formed by dehydration synthesis, so by removing a water from the molecule to help break them apart. So by breaking them apart, we're actually able to use those fatty acids as a potential energy source. So the primary function of the lipids is for energy storage. Our next type of lipid is a phospholipid. So a phospholipid is very similar to a triglyceride, where you have your glycerol molecule here. Here at the bottom we have our fatty acid tails. So unlike a triglyceride, instead of there being 1, 2, 3 fatty acids, the last part of that glycerol is attached to a phosphate group. So there's your phospho part of this phospholipid. And then a lot of times your phosphate group will also have something else attached to it. So your phospholipids are very abundant in your body. They make up most of your cell membrane. And its structure for having the fatty acid tail, which is hydrophobic, and this region, which is the head region, being hydrophilic, actually comes into play to not only the structure of your cell membrane, where the hydrophobic regions will point to each other, and then your head regions will either point to the outside surface of the cell or the inside surface of the cell. So the last type of lipid I'm going to talk about is your cholesterols. So cholesterol is what's considered our parent steroid. So the steroids that circulate throughout our body as a hormone is made from cholesterol. And cholesterol can be synthesized in the body, and its levels are its synthesis or its creation inside the body is determined by how much cholesterol you take in the diet. So if you take in a bunch of cholesterol in your diet, then the cholesterol synthesis, which occurs mostly in the liver, will be reduced to balance out how much cholesterol you're taking in. Cholesterol is very important for your nervous system to function, as well as the structural integrity of all your cell membranes. So with your phospholipids kind of floating around in their little bilayer, the cholesterol molecules are embedded into that cell membrane to help maintain its structure. So keep it less fluid and a little bit more hardened in its structure. So other steroids, again, made from cholesterol are cortisol, which is our stress one, progesterone, estrogen, and testosterone, which are all our sex hormones. And then lastly, our bile acids, which are very important in digestion, which helps to break down the lipids from being larger fat globules into what's called emulsification droplets. So that becomes very important for AP2, particularly the respiratory and digestive lab exam. So again, bile acids help to break down fat into emulsification droplets. So there's only one kind of cholesterol. The different types of cholesterol, at least levels inside a particular molecule, give it its good and bad reputation. And it's basically due to the levels of cholesterol in lipoprotein complexes. So lipo for the cholesterol region, and then the protein being the rest part of that complex. So a high-density lipoprotein, or a HDL, has a lower ratio of lipid to protein and may help to prevent cardiovascular disease. So later when we talk about digestion, when cholesterol is being removed from these lipoprotein complexes, and those complexes now have very low levels of lipids in them because those lipids have been used up by the cells, then those complexes can be converted by the liver in order to pick up free cholesterol that's circulating throughout your bloodstream, and therefore helping with supposedly keeping your bloodstream at least clear and preventing of plaques. The other type that's considered your bad cholesterol is your low-density lipoproteins, which have more lipid to protein and is considered to contribute to cardiovascular disease. Okay, moving on to protein. So earlier I mentioned protein is made up of amino acids, which are the monomer building blocks to make up a protein. A amino acid is a central carbon with attachments. So one attachment is the amino group, and that's the same across amino acids. The other attachment is your carboxyl group, which is the same across all your amino acids. And then the last one is your radical group, and that determines what type of amino acid it is. So in the body there are 20 amino acids. Some of them are considered essential because they have to be taken from the body. They cannot be made by, cannot be synthesized inside the body. They have to be taken in through through your diet. So here's some examples of your different amino acids, where this section, amino group, central carbon, and your carboxyl group are all the same. And then this pink section are your radical groups that determine the differences in the different amino acids. So in order to put your amino acids together, they're put together by peptide bonds. So peptide bonds join amino acids together to form proteins. And depending on the number of amino acids that are in that growing protein chain determines its naming. So again, with your number going with the different names, so di for 2, tri for 3, oligo having a few, poly having a little bit more many, so that one's actually 15 is this number. And then lastly, proteins having more than 50 amino acids in its chain. So there's 4 levels of protein structure. You have your first level, which is your primary structure. So this is your one individual long chain of your amino acids. Then the next level is your secondary structure. So this one chain can either be spiraled into what's called a alpha helix or folded into what's called a beta sheet. So kind of like a zigzag pattern. Your third level is your tertiary structure. So it's a combination of straight sections, alpha helices, and then also, for example, here you have some beta sheets. So it's a combination of different types of folding for your protein, which is your final level that a individual protein can have. Now, some of your proteins will work together and form an even larger molecule, which is your quaternary structure. So that here, and one example of that is hemoglobin. So we have 4 individual proteins, and they're put together to form this hemoglobin molecule. So there's 2 alpha chains in purple, 2 beta chains in gold, and on the center part of each of these protein is your heme group, and that's where your oxygen will bind to. So in the center part of that heme group is also your iron. So that's why you need iron part of why you need iron in your body. So this quaternary structure is made up of individual proteins that will form kind of like a megastructure. So some of your proteins can have what's called a prosthetic group. It makes it a conjugated protein. So for example, in hemoglobin, the heme portion, so this portion here, that attaches to the protein. So it's not made up of amino acids, but it's made up of other molecules. That is a prosthetic group, and it just helps with the function of that protein. So the job of your proteins, you have structure, so the keratin in your hair, skin, and nails. Collagen being in, like, for example, your skin, so keeping it nice and stiff, if you will. Versus kind of getting wrinkly and saggy. In your bones as well as your teeth. Necessary for communication. So your hormones that travel throughout your body are made up of, some of them are made up of proteins. So it becomes the signal, which is the ligand, and then those ligands will bind to a receptor, and your receptors are usually also proteins themselves. Involved in membrane transport, so inserted into that lipid bilayer that makes up your outside layer of the cell. Those proteins that are embedded in there can either make a channel, like a tunnel, into the cell, or to carry something from the outside or inside of the cell to the other side. And another function is catalysis, so being enzymes that help to speed up a reaction. The next job, recognition and protection. So on the outside surface of your cell are conjugated carbohydrates. So the protein portion of our glycoproteins are necessary for our immune system to recognize that our cells belong to our body. So this is how it determines whether or not there's, for example, maybe a bacterial cell that's going around. Its glycoproteins are going to be different than the glycoproteins in your body. Also, part of your immune system are your antibodies, and so they're targeting to a particular pathogen. So this is why you get those vaccination shots, so that it can trigger a fake response to kind of a, for example, when you get like your flu shot, it's an inactivated form of the pathogen. So you still get that immune response, even though you're not actually having an active infection. And so when you still have that response, you're still building up the protection just in case you get exposed to that particular pathogen. They're also used in moving stuff across the cells. So using these cells' cytoskeleton to change shape and move along stuff throughout inside the cell. They also can help bind cells together. So we'll show that next chapter, showing how the proteins can help keep your cells together so you can form tissues. So with your proteins, with their job of being a catalyst, one category of a catalyst is enzymes, and it helps your reactions to occur very rapidly at body temperature, so you don't need to increase the body's temperature to help that mixture or chemical reaction to go faster. So your enzymes will work on what's called a substrate, and the naming of your enzyme is based on this substrate, and then you have this ase suffix at the end. So the enzyme amylase, which we'll cover again in the digestive section for AP2, amylase digests your starch enzyme. Definitely need to know this for AP2 as well as that lab exam. So the enzyme breaks down starch, which is another way of saying starch is amylose. So it's ose, it's sugar. Another way for saying starch. So amyl, that part of that group, and then the ase part is for telling of that it's an enzyme. So the job of enzyme is to lower the activation energy, which is the energy needed to get a reaction to start. So here in our little charts, we have the normal path of energy needed to get a reaction to go, and then here's the level of energy in that reaction. In the second chart, we have a smaller amount of activation energy. So because we have that enzyme, our activation energy is lowered, and therefore you don't have to put so much energy into the system in order to get the reaction to go. So here's an example. We have our substrate binding to our enzyme to form the enzyme substrate complex. Because the enzyme puts everything in close proximity to each other, and in this case kind of works as a lock and key because the enzyme is very specific for the substrate. So by putting them together allows for that enzyme to either work on the substrate or at least put them in close proximity for example, if you're putting them together. But if you're breaking them apart, still holding them in place where the enzyme can work on them to break apart into those different products. So for example, your enzymes that help with your digestion to break apart those nutrients into molecules that are small enough for our body to absorb. So again, your enzymes can be reused in a reaction. They're not consumed. So they might go through a conformational change as they're binding to or releasing the substrate and products, but the enzyme is not used up in that reaction. It helps to speed up greatly the reaction, so giving it astonishing speed to speed up a reaction to keep going. But because the enzyme is a protein, temperature, pH, and other factors can change the protein shape and therefore the enzyme shape, and therefore affect its ability to continue to function. So next semester we'll talk about digestion. So here we have two different enzymes. Salivary amylase, which breaks up starch inside your mouth. Inside your mouth has a neutral pH, and so salivary amylase works best at a pH of 7. Pepsin, which is an enzyme to break down protein, works inside your stomach, and so it works best at a pH of 2, being a very acidic environment that with all that hydrochloric acid, and so it functions best at that pH. So for example, when you swallow and some of that salivary amylase, which is in your spit, as you swallow it down and it reaches your stomach, it's no longer going to be able to work because it's at the wrong pH. Pepsin, once you partially digested your food in the stomach and then it moves to the small intestines. Earlier we talked about your pancreas making sodium bicarbonate. So that sodium bicarbonate neutralizes all that food and digested bits and particles. So the pepsin will no longer work in the small intestines because the pH is incorrect. So for your enzymes, again, optimum temperature for them to work is 37 degrees Celsius, something you need to remember for AP2, especially that lab exam. That's your temperature because that is body temperature, and so body temperature is your normal functioning temperature for these enzymes. Okay, last bit talking about the Last type of molecules, which make up your DNA and RNA, as well as ATP, which is your major energy molecule for the body. So what makes these up are called nucleotides. These are the monomers that make up your DNA, RNA, and make up your ATP molecule. So there's things that are common across all nucleotides, which is that somewhere it's one of those questions where in your study guide where it says the differences between DNA, I think I said DNA molecules or something like that, slightly off. What I meant was it should be differences in your nucleotides. I believe so. So there's, of course, you still have to know the difference between DNA and RNA too. But so between your nucleotides, what's common is your nitrogenous base and your sugar group. And then they also have a phosphate group that make up your nucleotides. The nitrogenous base is what's different between your different nucleotides. So the sugar and phosphate group's the same. So for example, if you're talking about DNA, all that's going to be the same. If you're looking at the stuff for RNA, then the sugar is different between your DNA and your RNA. So nitrogenous base is what's different between your different nucleotides. So one important molecule, which is our energy molecule in the body, is called adenosine triphosphate, shortened to ATP. So ATP. So the nitrogenous base for that is adenine. Then it has a ribose as its sugar, and then attached to it, instead of just one phosphate group, it has three. So there's your triphosphate for that one. So energy is stored in this bond between the phosphate groups. So if you take this ATP molecule, you break off one of those phosphate groups, you're going to release energy, and their body can use that. Another way the body uses the ATP molecule is by taking off two of those phosphate groups and forming what's called cyclic AMP. So it's cyclic because it goes in a circle here. So that phosphate group kind of rebinds to that ribose sugar. And then so it's monophosphate. So there's our MP. And this is usually used as a signaling molecule in the body. So the enzyme that is used to break off the phosphate group off of ATP is called adenosine triphosphatases. So again, earlier talked about enzyme naming, ACE for enzyme, and what's working on is the previous part of that word, so the adenosine triphosphate. So our ATP ACEs is what it is. Separates ATP into ADP, so diphosphate, with the free phosphate group, and then releasing energy by doing so. So the opposite case is phosphorylation. So it's the adding of phosphate groups. So if this were a chemical equation going back in this direction, so adding a phosphate group to, for example, a ADP molecule, making it into an ATP molecule. So the enzyme that does that is a kinase. So the job of a kinase, it actually can help to add phosphate groups to other molecules to either activate them or deactivate them, so causing a conformational change in those proteins. So next semester, or next for your AP2, we'll go into detail the production of ATP in the mitochondria. So we start off with glucose, and it goes through what's called glycolysis, and in that case, you'll make two ATP molecules and end up with pyruvate as your final product. Now, depending on the level of oxygen inside the cell, your cell can then take this pyruvate and ferment it into lactate, and only making two more ATP. And we'll go through that next semester on how those details work. Or if there's enough oxygen in the body, instead of this pyruvate being fermented, then the pyruvate is transported into the mitochondria. Again, here comes our oxygen, going through a big long process that we'll go into the details next semester, but eventually at the end being way more efficient in creating 36 molecules of ATP. Okay, so our last nucleotide polymer are our nucleic acids. and they have different jobs, which will go in more detail in the next chapter. But most obviously well known is our DNA, or our deoxyribonucleic acid. So DNA contains millions of different nucleotides, so very long chains. Some of the chain parts creates what's called genes, which are our instructions for making our different proteins. So we'll go into that later. And the other type is RNA, and there's 3 types of RNA. So it's ribonucleic acid. So the difference between them is the sugar. So in deoxyribonucleic acid uses deoxyribose sugar, and ribonucleic acid uses ribose sugar. So going back to RNA again, 3 types. You have your messenger RNA, so it's a copy of your gene. Your ribosomal RNA, which is necessary